All right, hello everyone. We're going to talk about cell communication today. This chapter is going to be a chapter between like the cell membrane chapter and the next chapter going forward about enzymes. It's not technically in the old book that we were using, but it is in the new book. They added this into the curriculum years and years ago, but basically what this is looking at cell communication and how different organs and different cells are going to respond in different ways due to chemical signaling within the body. This allows your body to, or I should say, animal bodies to respond due to changes in their environment. So let's start going over the first section here. First section here is just on general cell communication. We have our before we get to our three types, just the whole just idea of cell communication, just like I said, is you're sending and receiving signals to create some type of response due to changing environments. And this allows us to survive, or allows organisms to survive more effectively, because they can change their internal homeostasis, whatever, to their external changes. Okay, first type. We have cell-to-cell communication. You can see here, this type of communication is where the cells contact each other. We can see the cells contacting those plasmodesmata and the gap junctions, and they send across signals. You can also have cell membrane proteins touch, and that's another type of cell-to-cell communication. It's also called juxtacrine. I always talk about how these are like a paper note, where like if you want to pass a note to somebody, you have to actually physically be next to them to pass that note. You guys probably don't pass notes because you have cell phones, but when I was in high school and in middle school, we didn't have cell phones, so well, most of us didn't. So we had to pass notes, and this is how we communicated. Short distance communication, this is also called paracrine or synaptic, based on which what we're looking at. So you can see here this cell is just secreting certain molecules to target cells that are around. You can see the nerve cells also doing the same. This is like sending a Snapchat where it's going to go to a specific receiver. So you are still within the vicinity, but you're sending it to a specific receiver, and only that target receiver or that target cell will respond to that to that message. And lastly, there we go. We have long distance communication. This is where you're going to have, again, this is going to be through your circulatory system, or another type of system, but this is basically where hormones or different molecules are going to travel throughout the system, and it's going to target certain organs. This is the analogy here is like a Facebook post, where you can put this Facebook post up, but certain people will only respond in certain ways. So you might have some people like your post, some people comment, but different people are going to respond differently to this post. Some won't respond at all. And that's just like the endocrine system, or the using hormones to create some cellular response. So those are the three main types of communication that we're going to see in the body, and they are all based on distance they travel. So it's not really about the molecules; it's about how far the molecules have to travel. All right, the next section of cell communication is going to be on signal transduction. Now, I don't have these slides organized by like 5.1, 5.2, 5.3, because this chapter is just it's not in the book, and there's really it's kind of hard to flow. These first two slides, or these first two sections, are kind of organized, but after this is kind of just random. So I understand this is like the second day of information starting here. So signal transduction, this is the actual process where we're going to go from a signaling molecule to some response within the cell. So again, messenger molecule is going to bind to the receptor on a cell membrane. We can see we have it's called a ligand. This is a chemical molecule that bonds with the proteins. This can't pass through the membrane usually. We'll talk about that later later on, how that can maybe change. But we have a sending molecule. You can see there's a ligand. This is some molecule. This is usually not a protein, but this molecule is going to dock and or bind with the receptor protein. Again, these are membrane proteins in the membrane. We have a sending cell, and we have a target cell. You can see this non-target cell has no receptor for a ligand. So not all cells will have the same receptors. A lot of them will have many different receptors, and they will respond in different ways. Again, in the whole goal of cell communication is to create some cellular response in some cells versus other cells. So again, these membrane proteins are called receptors, and these molecules are called ligands. Let me go back. Let me go back. There we go. Here's another way of looking at it. We have our ligand bonding with our receptor. It's going to activate other intracellular secondary messengers, and you're going to get some response. You can see these are three parts that we're going to go over. We have our signal perception. It's just like the signaling. This is just the initiation of the signal. This is the reception of the ligand. You have your intracellular signal transduction. This is the activation of other messengers within the cell. And then you have some cellular response. These are the three main steps of signal transduction. So just like I said, the first is the reception. This is where the ligand is going to bond with the protein. What usually happens is the protein will change shape in some way. And when you change shape, you change function. Good job. So when that protein changes function, it's going to activate usually some other relay of messenger proteins, and they're going to activate each other. So this molecule is going to activate this molecule. This molecule is going to activate this molecule. This molecule is going to activate this molecule. Almost like a game of, oh, now I'm forgetting it. It's like the mousetrap game where one thing activates the next. So again, that's the second step. This is the transduction of the molecule or the message. You can see again, this is the first part, the reception, molecule binding to the receptor protein. It's going to change shape and activate some secondary messenger. This transduction is the second part of signal transduction. And the last part is the response of the cell. So it's going to do something in response of this secondary messenger. There are many different responses within a cell, but this is going to create some response. And this is our third part of the signal transduction. Another way of looking at it, again, no ligand, nothing is activated. So again, we have that ligand, it activates this protein, which activates this protein, which activates this protein, which activates this protein, which turns on some target response in the cell, and you get something to respond. Either a cell goes through mitosis, or DNA is replicated, or you produce more ATP or something. This is your body controlling which cellular responses are active at different times. All right, this is the end of the second set. All right, so this is the third day of notes for cell communication. So we're going to be talking about how the cell has specificity or can just trigger certain responses that are very particular between the different types of cellular responses. Okay, so with that being said, let's just dive into it. So it says the same signal can trigger different responses depending on the receiving cell. So we can see here we have two different cells, and they are both being triggered by this specific ligand, but you can see their relay molecules inside the cell are different, and they're going to create different responses. So just because one cell has a response to a particular ligand, it doesn't mean all cells will have that particular response. I always like to talk about adrenaline at this point. During class, I show a scary, like a jump video, and I get, the kids to understand what adrenaline feels like, and you just go to that relation to, okay, so this is what adrenaline does. It makes me breathe faster. It makes my heart beat quicker. It makes me sweat sometimes. It breaks down glycogen. So there's a lot of different responses that one molecule will have based on the adrenaline binding with the reception of different cells. We have this and or kind of way of looking at different responses. You can see here, both of these are going to be needed for a response. So it's, again, both of these are needed. And this one, it's either or. So as long as we have one of these, you can get a cellular response. There's literally thousands of these different type of responses. And I kind of relate this, teaching about cell communication as like, hey, there are just tons of different cars. And we're going to look at, different cars, individually, but it's really hard to talk about all these different cars because there's so many different types and so many different, modifications and changes and differences. So I'm just generalizing a lot of these different cell communication pathways. A couple molecules that we need to know about are kinases and phosphatases. Kinases are enzymes, again, this is an enzyme, ends in ase, that transfer a phosphate group, usually from ATP to another molecule. So you can see here we have our protein kinase. It is going to, sometimes it will activate. You can see here it's using ATP and it's bonding a phosphate group on there. And then the phosphatase will actually remove that phosphate group. So kinases add a phosphate group, phosphatases remove the phosphate group. Now, sometimes that phosphorylation will activate it, sometimes it will deactivate it. Again, it depends on the specific type of cellular response or signal transduction. But understand, kinases add phosphate groups, phosphatases take them off. Phosphorylation cascade. This is where you're going to have a series of kinases that are going to add a phosphate group onto another kinase, activating it. That activates another one. This is kind of like a bucket brigade. So you can see here, this kinase activates this kinase, activates this kinase, and then this activates some other protein that's going to create some cellular response. There is a It's all, again, almost like a bucket brigade, but there's a reason why we have these phosphorylation cascades. I love these memes here. Yep, these kinases give away phosphate groups. What this allows us to do, or allows the cells to do, is create a amplification. So we have one ligand bonding to this receptor, which activates this, protein, that activates these, that activates these, that activates these. And what's going to happen is called this amplification. So we can have a small amount of ligand that creates some huge cellular response in the cell. And the reason why we would need that is because of evolutionary thinking. You want to be able to respond to your environment very quickly. If there's a fire, if there's a predator, if there's something going on that you need to respond quickly to, in case if you don't, you would die. We want to be able to respond to these molecules or these changes in environments very quickly. I'm not going to show this video. All right, so in diagrams, what you're going to see is something called arrows and bar-headed lines. Usually arrows, again, this is not all the time, but usually arrows mean they activate something or they're moving. So you can see we have some arrows here. So you can see these two molecules look like they activate this. This goes into this. Sometimes they either change. There's not a lot of guidelines or rules about these, but generally arrows show moving or activating molecules. Bar-headed lines, these are called bar-headed lines. These usually show that a molecule is being inhibited. So you can see here, pKa looks like inhibits this process. We'll get some practice with these a little bit later on in this chapter, but that's just a generalization for this, for cell communication. Cell communication disorders. So these are just different disorders that are created by some type of signaling pathway not going right. Either the ligand's not produced, the receptor protein doesn't bind because that's the wrong shape, or there's secondary messengers that aren't being produced in the right way. There's a lot of different types of cell communication disorders, and there's just a lot of different types because, again, there's so many different ways that we regulate our responses. There has to be a lot of also ways that these can go wrong. So think of like a car. If one thing goes wrong in a car, there's a host of other things that will go wrong because of that one thing. If your computer's bad, if your gas lines break, or if your engine just goes, there's a lot of different things that can go wrong, and so they manifest themselves in different ways. So these are called cell communication disorders. All right, intracellular receptors. I think this is going to be the last slide for this. Yeah. Actually, I think this is the last slide. Cell communication disorders is the last slide. So this is the end of day 3. This is going to be day 4 of cell communication. We talked about how the ligands in the first chapter, or the first part of this chapter, have to bind with a membrane protein to initiate some signal transduction within the cell. Well, not all cell communications will bind with that membrane protein. Some will actually cross the membrane, and these have to be either small or hydrophobic because, again, those membrane proteins, the membrane separates the outside from the inside. Those molecules are going to get into the cell because they're small, they're hydrophobic, or nonpolar. But anyways, so we have these intracellular receptors that bind with the ligand, and they can create some type of intracellular response, and they usually activate DNA to produce some protein. You can see here we have our extracellular fluid, our hormone that's going to bond with a receptor. It activates this receptor protein. It's going to activate DNA to make mRNA, which activates a protein to produce, ribosome to produce that protein. So this is just another type of intracellular receptor. Receptors on the plasma membrane. Yep. Okay, so there's 3 different types that we're going to kind of briefly go over. We have G protein coupled reactions or receptors, receptor tyrosine kinases, and ion channel receptors. And there's a lot of words there. There are 3 basic types of receptor proteins that we're going to go over, and the first one is the G protein coupled receptor. Receptor protein binds with a ligand, so you can see here's our ligand bonding with our G protein. The G protein is going to activate something called GDP into GTP. This is going to activate another enzyme, and then that enzyme creates some cellular response. All you have to know is this is just one type of cellular protein that's going to bind specifically and use GTP and GDP. Here's a little video of it. You can see here we have our ligand bonding with the receptor. It's going to use GDP and GTP to activate another protein. This activates another protein, and it's going to keep activating other proteins to create some cellular response. Not much is need to be known here. Tyrosine kinase receptors. These are membrane receptors. They are going to bind with 2 ligands. You can see here what happens. These 2 ligands bind. They are going to activate the protein to bond with each other. they are going to be phosphorylated, and then those phosphorylated tyrosine amino acids are going to create some cellular response. I know, again, this is very difficult. There's a lot of working parts here, a lot of words, but basically what you're going to see here is these two ligands bond with the tyrosine receptors, and they're going to, there we go, bond, come together. They are going to activate and become phosphorylated, and then they create some cellular response. Last one's the easy one. These are ligand-gated ion channels, where again, a ligand is going to bind to a ion channel. It's going to change shape, allowing the entrance or exit of certain ions or certain molecules, certain whatever. And that's just going to be a way for the cell to regulate what goes into and out of the cell. This is day 3 of the, day 4 of the notes. So you can see again, we have this molecule binding here. It should allow at some point, here we go, the passage of those molecules through. Last section in cell communications on feedback loops, and we have positive and negative feedback loops. This is where I'm going to focus here on the positive feedback loop. This is where the product of the reaction is going to lead to an increase in the reaction. Basically what this means is the product B is going to stimulate A. So when there's initial stimulus of A, the product, or it's A is going to make B, B is going to stimulate for more A is then going to make more B stimulates more A, and this is a positive feedback loop. This is where we create more B to create more A to create more B, and just keeps on creating more and more of each other. Negative feedback loop. This is where A is going to stop or be stopped by B. So you can see here the product returns condition back to its stable point. A produces B, B stops A, so A stops producing B. Great example of a negative feedback loop is your body temperature. So like when you get too hot, you're going to start to sweat, which cools you off. So again, it's going to stop that process. When you cool off, it's going to stop that process of sweating. Or when you get too cold, you're going to shiver, which again stops, or it's going to increase your heat, which then stops that process. So it's a way that we maintain homeostasis. Okay, this is the end of the chapter.